Hello, and welcome to the second part of our episode for cell division. I am Sir D, ang inyong science terrific teacher ng grade 8. Last episode, we have talked about the importance of cell division and mitosis. For this part, we are going to focus on the cell division that happens in our sex cells called meiosis. Almost all of our cells in our body are considered to be somatic cells. These cells continue to grow and divide through mitosis. But as an individual reaches puberty, the sex cells called gametes develop further due to hormones produced by the reproductive system. As a result, it will undergo cell division unique from somatic cells. Meiosis is a type of cell division that is specific to germ cells. Gametes are sperm cells for males and egg cells or oocytes for females. Gametes are haploid cells. A haploid cell contains only 23 chromosomes, half of the complete set of 46 chromosomes. Somatic cells usually have diploid cells, which has the complete 46 chromosomes. In order to complete the 46 chromosomes needed by gametes, 23 chromosomes will be coming from the sperm cell and 23 from the egg cell. During fertilization, the complete number is restored. There are two stages of meiosis. We have Meiosis I and Meiosis II. Let's now talk about what happens to the gametes when it undergoes meiosis. Let's start with the first half of the process. Meiosis I is known as the reduction stage. This stage is divided into Prophase I, Metaphase I, Anaphase I, and Telophase I. It will start with Prophase I. During this stage, the germ cell and the chromosomes are already duplicated. A homologous chromosome pair forms a tetrad, which is composed of 4 chromatids. The nuclear envelope breaks down and the spindle microtubules are formed. The chromosomes fuse with their homologs in a process called synapsis. Homologous pairs can be seen. Similar to prophase of mitosis, homologous chromosome, each with 2 identical sister chromatids, will condense. Crossing over can happen during the last part of this stage. What are homologous chromosomes? These are similar in chromosomes, but they are not identical. Each homolog has the same gene in the same order as the other homolog, but the alleles for each trait may not be the same in the homologous pair. What about crossing over? Crossing over is the process wherein homologous chromatids exchange genetic material to ensure greater variety in gamete formation. After that, homologous chromosomes line up randomly on the equatorial plane and they separate independently. Their separations result in 2 nuclei having half the number of chromosomes. This process will result in variation of traits among the daughter cells due to the recombination among the genes of homologous chromosomes. After Prophase I, it will then move to Metaphase I. During this stage, the spindle fibers attach to the homologs at the kinetochore. The tetrads are aligned at the center equator due to the tension built from the spindle fibers. Next, we have the Anaphase I. What happens next is the chromosomes that make up the homologous pair separate from each other, and they start to migrate towards the opposite cell poles. Their positioning and direction are in random, so it doesn't matter which of them moves towards which side of the pole. Lastly, it will move to Telophase I, wherein two daughter cells are produced. The cleavage forms, thus cytokinesis begins. Each daughter cell contains only a haploid number of the chromosomes. This is where it gets different. After meiosis I, it will undergo another stage of division called meiosis II. This includes prophase II, metaphase II, anaphase II, and finally telophase II with cytokinesis. Meiosis II is a division stage similar to mitosis, except that the cell does not undergo interphase anymore. This division maintains the haploid number of chromosomes and completes the goal of meiosis to produce 4 genetically unique cells from one parent cell. Thus, meiosis reduces the number of chromosomes and creates genetic diversity. During meiosis II, it will start with prophase II. Under this stage, it will begin with no more replication of the DNA in the chromosomes. The nuclear envelope breaks down and the spindle microtubules form. The centrioles duplicate and then separate into 2 chromosomes. Sister chromatids of each chromosome are still attached to the centromere. After prophase II, it will now move on to metaphase II. During this stage, each daughter cell is now attached to the fully formed spindle. In contrast with metaphase I, single chromosomes instead of homologous pairs are arranged on the metaphase plate. Each chromosome of the sister chromatid is attached to the kinetochore, microtubules from the pole and faces the opposite poles. After which it will move to anaphase II. The centromere separates and each individual chromosome from the sister chromatids migrates to the opposite cell poles. Lastly, telophase II will commence. In this last stage, a nuclear envelope forms on each set of chromosomes. Cell division is now complete. Cytokinesis takes place from forming 4 haploid cells, or single-stranded chromosomes. Due to the crossing over, some chromosomes are seen to have recombined segments from the original parent chromosomes. To sum up our lesson, we will look into the major differences of the 2 types of cell division. First is the type of cell these processes occur. Mitosis happens in somatic or body cells, while meiosis happens in the gametes. What about the number of cells produced at the end of each process? In mitosis, there are 2 diploid daughters produced. These daughter cells formed are genetically identical to the parent cell. On the other hand, there will be 4 haploid daughter cells produced at the end of meiosis. These haploid cells will carry the traits from both parents. In multicellular organisms, mitosis results in growth rather than formation of new individuals. Both processes is significant for the existence of life. Mitosis in most organisms is purposely for growth and repair. However, among unicellular organisms, mitosis is a means of reproduction. Meiosis, on the other hand, plays a vital role in the production of correct number of gametes. This is essential for reproduction, as well as for the uniqueness of organisms. This process of cell division may be a brief process, but they play a vital role in the existence and continuity of life on Earth. This closes our lesson for this episode. Always bear in mind that you are unique and no other person looks, thinks, and will be like you, thanks to cell division. This is Sir D, your grade 8 superb science teacher, bidding you farewell, and I'll see you next time.